the components of colorimeter and use of colorimeter. Okay. So, the principle of photometry is Beer-Lambert's law, is dependent on Beer-Lambert's law, and we will learn about the terms absorbance and transmittance. From the derived equation we will learn how to derive the concentration of an analyte, and we will know about the components and applications of a colorimeter. Okay. Acha. Now coming to the general introduction. From the word photometry, we can say photometry is nothing but measurement of light. Photo and metry, measurement of light, and colorimeter is, or colorimetry is, measurement of color, colors. Colorimeter, colori, not calorie. Okay, COLORI. Okay, colorimeter. So color or color comes, that means color, and metry means measurement. Okay, so what is like light? Light is an energy, form of energy, and what is color? Color is the reflected lights in the visible spectrum. So what is the visible spectrum of light? Wave diode, that is 400 nanometer to 700 nanometer. Above 700 nanometer we call it as infrared, and below under 400 nanometer it is ultraviolet. So human eyes can appreciate or can see only this wavelength. So that is why we can see colors according to different wavelengths of these light spectrum wavelengths. Okay. And from that we can easily say why a flower, red flower is seen as red and a green leaf is seen as green. Absorb, reflects. So the red flower reflects the red wavelengths and green leaf reflects the green wavelengths. All other wavelengths are absorbed. Clear or not? Now moving to our photometry. So suppose this green test tube contains this test tube is containing a green solution. Okay, and we are passing visible light spectrum. So what will happen? The green solution will reflect green. So green is reflecting. Now all others should be absorbed, but some is getting emitted also. Okay. So from this, other than the reflecting part, we can get two other parts. One is the emitted part, one is the absorbed part. Some amount of the wavelength is absorbed in the solution, some is reflected and some is emitted from the solution. When a visible light is passed through a solution, here it is green solution, that is why green color is getting reflected. Okay. So if the intensity of incident light or the original light is I0 and the transmitted light intensity is IE, we will denote transmittance as IE by I0. So transmittance is the ratio of emitted light and the incident light. Okay. How much of the incident light is emitted is known as transmittance. And transmittance depends on which factors? Nature of light absorbing substance, that means what is in the solution, what type of solution it is. Number 2, concentration of substance. Okay. How concentrated the substance is, the more concentrated the substance will be, the more it will absorb. Okay. Next, the wavelength of light. Which wavelength we are passing? In this we are passing all the visible wavelengths. But if we pass only green, it will be reflected. Right? And length of path, that is the solution length in which the light is passing through. Okay. Now coming to the laws. There are two laws of photometry, Beer's law and Lambert's law. Okay. So how to remember? Lambert's law deals with length, path length, and Beer's law deals with concentration of the substance. So can you see here 3 test tubes, they have different color intensity of the solution. First one is very light, second one is medium and third one is darker. Okay, and see the emergence lights, how much it is emitted and how much is absorbed. The more the concentration is, the more is absorbed and lesser is emitted, taito? And color intensity is directly proportionate to the concentration of the substance. If we add much color or much substance, it will create darker intensity solution. বোঝা যাচ্ছে যে আমি যদি একটা সলিউশনে অল্প কালার মেশাই বা কালারিং এজেন্ট অল্প মেশাই, the concentration of the coloring agent is directly proportionate to the color intensity of the solution. So here in the light colored solution, concentration of the colored substance is low. In darker and much darker, the concentration is increasing, that's why the color intensity is increasing. Along with the intensity increase or concentration increase, what else is increasing? Absorbance. The power of absorbing the light is also increasing. That's why emitted lights are getting low. Darker the solution, emitted lights are low. বোঝা যাচ্ছে? In darkest solution, see, only one arrow is getting emitted, whereas in the light solution it was emitting 3 arrows. So with increasing concentration, transmittance is getting decreased. So what Beer's law states, the amount of light transmitted through a colored solution decreases exponentially with increase in concentration of the colored solution. As we increase the color intensity of a solution or the concentration of the solution, transmittance will be decreasing exponentially. That is Beer's law. So in the terms of absorbance and transmittance, we can say with increasing color intensity, concentration will be also increased and absorbance will also increase, whereas transmittance will be decreased. Clear? And we can write it like this: concentration is directly proportionate to absorbance and it is indirectly proportionate to transmittance. বোঝা গেছে? And absorbance is also denoted as optical density. Okay, optical density. It is related with concentration. In normal terms, density is related to concentration or not. So optical density is how much light is passing through or how dense is the solution optically. So absorbance is known as optical density also. Now coming to Lambert's law. Acha, in case of Beer's law, one thing, look at the test tube diameters, they are same. The test tube diameter is the path length of the light. Light is passing through the solution, and how much is the solution length? That is the diameter of the test tube. So here in Beer's law, we keep the path length constant. Okay, so here all the test tubes are of same diameter, only concentration is increasing. But in case of Lambert's law, what happens? The path length, it is relation of the transmittance with the path length or absorbance with the path length. So we have to keep the concentration constant here. See, in 3 test tubes, they have different diameters, but the concentration is same. The color intensity is same in all the 3 test tubes, but it differs in the diameter. Taito? So in first test tube, the diameter is lowest. That's why emitted lights, they are more. As the diameter is increasing, the emitted lights are decreasing. So here what happens in Lambert's law? Lambert's law states the amount of transmitted light decreases exponentially with increased path length of light in the solution through which light passes. Okay, if the test tube diameter increases, the light transmitted through the solution will be decreased. Clear? Boja geche? So Lambert's law deals with path length. Easy to remember, L for length, L for Lambert's, and Beer's law deals with concentration of the substance in the solution through which light is passing. So in Beer's law, concentration is directly proportionate to absorbance or OD and indirectly proportionate to transmitted. The more concentration, the less transmitted light. In case of Lambert's law, what happens? As the pathway increases, the transmission decreases. Clear? So here it is the equation for Lambert's law, or how we can write it. Path length is directly proportionate to absorbance and indirectly proportionate to transmittance, where the concentration is kept constant. Concentration in both, all they are same. Now by combining these two laws, in photometry we combine both the laws. So we call it as Beer-Lambert's law. Okay. So in Beer-Lambert's law, what we take a fixed diameter test tube. By keeping the length constant, we inactivate the Lambert's law, but they are combined law. Okay. Beer-Lambert's law. We are keeping the length, path length constant by keeping a fixed diameter test tube. So here the path length is constant, only the Beer's law is active, but we call it as Beer-Lambert's law. And what happens here? It deals with only the concentration of the substance. ঠিক আছে? আমরা কেন দুটো আলাদা করে জানলাম? Because absorbance or transmittance, they depend on multiple factors. I showed you in the first slide the type or nature of the substance, concentration of the substance, pathway, wavelength, all these affect these points. So তার মধ্যে changeable factor কি কি ছিল? What were the changeable factors? Light তো আমরা fix একটা করে দিতে পারি. We can pass a fixed wavelength. Path length chilo, so we have fixed it here, we have kept it constant and it is 1 centimeter. The test tube we use in colorimeter or spectrophotometer, it is of 1 centimeter diameter. Okay. Now only the concentration variation will change the OD or the absorbance. So from that change of concentration of known solution and unknown solution, we can easily measure the concentration. So OD or absorbance, that is equal to, we can write KCl, where K is the coefficient, absorption coefficient of a particular substance. Suppose we are preparing a glucose solution. We are preparing a glucose solution. So for glucose, in different concentration glucose solution, absorption coefficient will be same because the substance is glucose. Okay. But we have kept the path length constant. So OD or absorbance will be directly proportionate to concentration only. এটাকে আমরা কি করে দেখাবো? See, if we take two solutions, okay, one with known concentration, another with unknown concentration, and we estimate the absorbance in the machine. Okay, absorbance can be measured, OD can be measured in our colorimeters, okay, or spectrophotometer. So if we measure the OD of a known substance, that is standard concentration, and we are measuring unknown concentration substances OD, so we will get OD standard, OD unknown. We know the concentration of standard, we don't know the concentration of unknown. Can we find the concentration of unknown? How? Can we write this? K is same for the same substance. I told you for glucose, K will be same. L, that is path length is fixed. So can we say ODS is equal to CS, or can we write it like this? ODU by ODS is equal to CU, that is concentration of unknown by concentration of standard. Clear? Can we say this? From the OD of known and unknown, concentration of known and unknown, can we say this? And from that equation we can write this. Concentration of unknown will be OD unknown by OD standard into concentration of standard. And what is standard? Standard is the sample that we know that is a known sample. We know what is the concentration. Standard solution we can make it in our lab. Suppose we have prepared a solution of 100 milligram per liter glucose solution, okay, and we have measured the OD. Now I have provided you with an unknown concentration sample, which I also don't know what is the concentration. I have randomly prepared a glucose solution. You can measure the OD in the instrument. From that OD, what we have, what are the values that we have? OD standard we have, concentration of standard we know, that is 100 milligram per liter, and OD unknown we have measured from the machine. So by this equation, can we measure the concentration of unknown? Yes, clear? And for volume correction, like if the concentration of standard is given in milligram per milliliter, and we provide the report as milligram per DL, right? For our blood reports, we mainly provide the reports in milligram per DL. So for volume correction, we can do this next part. But the main equation is that, the second one, that is Cu is equal to ODU by ODS into Cs. Okay. We already know T is equal to IE by I0. Transmittance is IE by I0. What is IE? Emitted light intensity. I0 is incident light intensity. Incident light that was passing through, the original light that was passing through the solution, that was I0 intensity, and the emitted light intensity, that is IE. But we always write transmittance in percentage. Okay, মানে একটা লাইট আমরা পাস করাচ্ছি তার পার্সেন্টেজে আমরা বলি যে how much percentage is emitted. So it is written as like this, 100%. Percentage transmission is 100 into IE by I0. So from that we can say IE by I0 is percentage transmission by 100. Okay, and we also know that absorbance is inversely proportionate to T. A inversely proportionate to T. If we take log of that equation, we will get A is equal to minus log T. Okay, so A is equal to minus log T, we can replace T by IE by I0. T is nothing but IE by I0, right? So IE by I0, if converted in percentage value, it will look like this: percentage transmission by 100 minus log 100 will come up and it will be the value of 2. So A is equal to 2 minus log percentage transmission. Absorbance or OD, optical density, how much the solution is absorbing light. How much light is getting absorbed by the solution is known as absorbance. Okay, that is OD, absorbance. OD or optical density is equal to absorbance. That absorbance is inversely proportionate to transmittance. The more it is absorbed, the less it is emitted, right? More absorbed, less emitted. তাহলে inversely proportionate. তো সেটাকে আমরা যদি লিখি, আমরা log value করি, তাহলে A will be equal to minus log T. Why minus? Because 1 by T ছিল তো, A proportionate to 1 by T ছিল, তো log নিলে কি হয়ে যাবে? Minus T minus log T হয়ে যাবে. So T is nothing but IE by I0 and it is expressed in percentage value. T টাকে আমরা সবসময় percentage, like 90% is emitted. Transmission is 90%, এরকম করে বলা হয়. So the percentage value আমরা যদি এখানে put করি, so what can be the maximum absorbance? From this equation, A is equal to 2 minus percentage T, log percentage T. Maximum কত হতে পারে? Log percentage T এর lowest value কত হতে পারে? 0 হতে পারে তো? তাহলে absorbance can be maximum of 2. ঠিক আছে, মানে OD maximum 2 হতে পারে. ওকে, এটা ম্যাথমেটিক্যাল কোয়েশ্চেন, অত মানে মাথা ঘামানোর কিছু নেই, এটা ইকুয়েশন প্রুভেন। তোদের খালি এখানে জানতে হবে যে OD maximum 2 হতে পারে। ওকে, আচ্ছা এবার আমরা যখন when we perform a test in our lab, what are the required ODs we need, তাই তো? কি কি দেখ, ফার্স্ট অফ অল একটা টেস্ট করার জন্য we require a sample like the analyte which we will be measuring. We will need reagents, and to calibrate the machine we require distilled water. So why we require distilled water? Because the optical density of distilled water ideally should be optical density or absorbance. Absorbance of distilled water should be absorbance, the amount of light that should be absorbed in distilled water, ideally it is 0. 100% should be transmitted. Distilled water is transparent, clear, 100% should be transmitted. So the optical density value should be 0. ঠিক আছে? So by placing a distilled water in a test tube, we calibrate the machine. We calibrate the machine to its optical density 0. Okay? Whatever OD it is giving, we calibrate it as 0. Because ideally distilled water's OD is 0, because it should transmit the 100% light. Now see here, next step, in any reaction, it's not necessary all the analytes will produce a colored compound on dissolving it, তাই তো? What is the color of a glucose solution? Glucose solution এর color কি? So can you distinguish it from distilled water? No to. So we need to perform a reaction of that particular analyte to produce a colored compound. ঠিক আছে? So we will run a reaction in the test tube to produce a colored compound that will reciprocate the concentration of that analyte. মানে যতটা গ্লুকোজ থাকবে, আমি যদি একটা রিয়াকশন রান করাই, গ্লুকোজ ইজ দ্য সাবস্ট্রেট হেয়ার, it will produce the same amount of product as the substance. যতটা সাবস্ট্রেট দেব ততটাই তো প্রোডাক্ট তৈরি হবে, right or not? So the concentration of the colored product that tallies with the concentration of our analyte. So by measuring that colored solution's intensity, we will find out the concentration of this analyte. So to run the reaction, we need reagent. Reagent ki always transparent hoy na? Tell me one reagent that is not transparent, colored reagent you have worked with. Benedict reagent, another one sulfosalicylic acid reagent. So these are colored reagent. So if we place a colored reagent will also give an OD. Colored reagent নিজেই একটা absorbance দেবে তো। So to eliminate that fault or to eliminate that error, we have to measure the reagent blank. Okay? So blank is of two types. One is water blank that is used for calibration of the machine. Water blank, distilled water, we use for calibration of the machine, and reagent blank to eliminate the error. So reagent blank er OD we will denote as ODR. Now see, in standard solution sample, we know the concentration of the analyte, and we are running the reaction, we are doing the reaction with a known solution, known concentration solution. So that OD of that standard solution, in this solution the reagent is also there and the sample is there, তাই তো? So the OD of standard is the combined OD of reagent OD and standard solution OD. And same, the unknown OD is the combination of reagent OD and unknown solutions OD. বোঝা গেছে? So to be specific and to eliminate the errors, we have to subtract the ODR from both the ODs. ঠিক আছে? বোঝা গেছে? আমাদের সেই প্রথম রিঅ্যাকশনটা, প্রথম ইকুয়েশনটা, concentration of unknown is equal to OD unknown by ODS jeta chilo, we have to subtract the OD of reagent from both the ODs and after the volume correction. So this is the equation. বোঝা গেছে এই পর্যন্ত বোঝা গেছে photometry equation বোঝা গেছে what is absorbance and what is transmittance, what is the relation between absorbance and transmittance, inversely proportionate, and if equation by mathematical equation we express it will be A is equal to 2 minus log percentage T. Okay, and the percentage is it will vary between 0 to 100. Percentage of transmission, how much it will vary, what is the range, 0 to 100. So the value of OD can be 2 maximum and lowest can be 0. Now coming to colorimeter. So this is the colorimeter instrument, you all have seen it in our spotter room. Okay, so what are the external parts? External parts of the colorimeter we can see externally. First of all, there is a display, digital display, তাই তো একদম উপরে একটা ডিজিটাল ডিসপ্লে দেখতে পাচ্ছ, wheel. What is the use of the wheel? To change the wavelengths, তাই তো. So there are different markings for different wavelengths, we can put it in our desired wavelength. Here is a cuvette place, a place that where we can put the test tubes or the cuvettes. The test tube designated for colorimeter is known as cuvettes. Okay, and there is another button to start. And the internal components, another thing that is, I already told you, if we are measuring a colorless biochemical compound, we have to run a test that will give rise to a colored product, and from that we will measure the concentration of that analyte. Now these are the internal components or parts, internal parts of a colorimeter. So until this in photometry আমাদের কি কি লাগছে, what are the things that we require in a photometric experiment or photometric measurement, কি কি লাগছে? একটা লাইট সোর্স লাগছে, একটা সলিউশন লাগছে, we need a photocell. Photocell কেন? To capture the emitted lights. আমরা একটা বাল্ব দিচ্ছি for the source of light, একটা solution to pass through, then a photocell to capture the emitted lights, and then a display or a galvanometer, ammeter, whatever that is a display for the measurement. So here in colorimeter these things, these parts are in sequence. What are they? Light source, and then entrance slit for the light source and a condenser. Okay, so why entrance slit? Because if we put a bulb somewhere in a colorimeter, it will produce much amount of light. So for like separate a particular amount of light or a small, very small amount of light, we use a entrance slit, and a condenser, that is to make them parallel rays. Okay, so a minimum amount of light will enter through the slit, and then there will be a biconvex lens which will make them parallel. Okay, then comes a filter. What is the use of a filter? It is optical filter, not your filter paper. Okay, so optical filter will filter the desired wavelength only, and then there will be cuvette holding the solution, then there will be photocell, galvanometer for display. They are placed like this, in this order. Can you see a light source, then a entrance slit, then condenser, then filter, then cuvette containing the sample or the solution, then there is a photocell, and finally a display. What is the ideal light source? It should be a tungsten bulb that will produce the visible wavelength, light of visible wavelength. And what is the visible wavelength? 400 to 700 or 750 around, okay, nanometer. Now see, with the entrance slit only a specific minimum part of light is entering, and by the condenser it is converted into parallel rays. In case of Beer's and Lambert's law, two more things were to be specified, that we are using monochromatic parallel beams. Okay, monochromatic means only one specific wavelength, color wavelength, and they should be parallel. So here we have made them parallel by a condenser. Now we will make it monochromatic, or we will filter all the other wavelengths and will pass only a specific wavelength. That's why the filter is used. In case of colorimeter, the filter is mainly colored glasses. Okay, there is a wheel with different colored glasses, and we put that glass, by rotating the wheel or by rotating the like numbers, we put the colored glasses in front of the light source. So if we put a green colored glass in front of a white light, which light will be emitted? Only green, all other will be absorbed, and so on. Like if we place a yellow glass in front of a white light, only yellow light will be emitted. So by this we can make it monochromatic. Bojha geche? Clear? So and here is the cuvette. So there are few features of cuvette of colorimeter. It is also asked, write down two specific features for colorimeter cuvette. So the diameter is fixed, 1 centimeter diameter, and the thickness should be uniform. We don't use the normal test tubes in colorimeter, okay. There is designated 1 centimeter test tubes, and they are ideally made up of borosilicate glasses, and they should be, the wall should be free from scratches and any air bubble, because it is dealing with lights and it should not be refracted, reflected or absorbed by anything other than the solution. Okay, clear? Bojha geche? And as the light passes through the solution, it will fall on the photocell and the OD will be measured. Optical density or absorbance will be measured, and we use the complementary filter. Can you see, like the solution is pink in color and we are using a green colored wavelength. Why? To maximize the absorption in the solution. Green is complementary to pink. Okay, if we remove pink from white spectrum, it will be green. Okay, or vice versa. If we remove the green color from white spectrum, it will be pink. That is already provided in books. Okay, like this. We already know which color is complementary to which color. So according to our product color or the solution color, we adjust our filter, and we have to put the complementary filter for that color to maximize the absorption. Okay, যদি আমরা একটা পিঙ্ক কালারই ওর ভেতর থেকে পাস করাতাম তাহলে কি হতো? মোস্টলি উইল বি এমিটেড, তাহলে আমরা কনসেন্ট্রেশন মাপবো কি করে? সেই কালারড প্রোডাক্টটা কতটা লাইট অ্যাবজর্ব করছে তার উপর ডিপেন্ড করেই তো আমরা কনসেন্ট্রেশন মেজার করব। Depending on the OD of that substance or the solution, we will measure the concentration. So for proper or exact mathematical representation or value finding, we need to take a larger number. খুব small number পেলে তো সেখানে ভুল হওয়ার চান্স বেশি। আমি যদি একটা বড় ভ্যালু পাই সেখান থেকে আমার কনসেন্ট্রেশন বার করা easier. That's why we always use complementary colors. Okay, now coming to the last part, that is use or application of a colorimeter. So we use, like colorimeter is the primitive most photometric instrument used in biochemical labs. ঠিক আছে? Nowadays the photometric instruments are super modified. ঠিক আছে? In colorimeter we can get the OD only or optical density only. all the mixing reagent and sample, getting the product, then finding the OD, all we have to do manually in colorimeter. Taito, like we have to run a test outside the machine to find a colored product, then we have to put that in the colorimeter and we have to find the OD. We will get an OD and then we have to place the values in our equation and find out what is the concentration of unknown, because we know the concentration of the standard and from that we can find out the OD of standard, and we have already found the OD of unknown. So, from that equation we can find the C concentration of unknown. So, everything we have to do manually. The next modified instrument is semi-auto analyzer. Okay. So, in semi-auto analyzer it is a modified colorimeter with an internal processor. Okay. So, what happens there? We mix the reagent, run the test outside and we feed the machine. Machine will calculate the concentration on its own. একটা সফটওয়্যার প্রোগ্রাম অলরেডি ইনস্টল রয়েছে, সেখানে ও নিজেই ওডিটা বসায়, কনসেন্ট্রেশন দেওয়া রয়েছে, স্ট্যান্ডার্ডের কনসেন্ট্রেশন, স্ট্যান্ডার্ডের ওডি ওকে বসিয়ে দেওয়া রয়েছে। দ্য মেশিন ইটসেলফ উইল ক্যালকুলেট অ্যান্ড গিভ আস দ্য এক্সাক্ট কনসেন্ট্রেশন। So, we do not need to do the calculation part in semi-auto analyzer. After this, the next modified machine is full auto analyzer. So, in case of full auto analyzer, what happens? We place the samples, we place the reagent in designated places, machine will pick the sample, machine will pick the reagent, machine will mix it inside and it will run the test, perform the test and find out the OD, and then it will calculate the concentration and finally it will show us the concentration. ঠিক আছে? বোঝা গেছে তো? তাহলে আমাদের day to day life এ বা আমাদের ল্যাবে we use to measure glucose, urea, creatinine, proteins etc. from different samples, not only urine. এতদিন তো we were dealing with urine only, urine sample only. Now from now on we will move to serum samples, okay, blood samples, and in blood, in biochemical parameters, all the biochemical parameters mostly are derived from serum. Cells we don't need. ঠিক আছে? Cells are required in pathological part. They count the cell count, RBC count, WBC count and platelet counts, all these, and we do all the biochemical analysis. So, from now onward we will start performing glucose, urea, creatinine, protein, cholesterol, alkaline phosphatase and all these, okay. But in colorimeter basic tests like glucose, protein, these are done. Clear though? So, next class you will learn about, like, not in this next class, next week we will start with glucose estimation. Clear? Clear? Everything clear up to this? What is the principle of photometry then? Which laws? Beer-Lambert's law, and Beer's law deals with concentration of the solution and Lambert's law deals with path length. By combining these two laws we can say absorbance is equal to, what is the full equation? OD is equal to KCl. KCl to, where K is absorption coefficient, C is concentration of the solution, L is path length. And for our ease of calculation we keep the path length constant and it is 1 centimeter. And what is the relation between absorbance and transmittance? Inversely proportionate. Okay, and what is the equation, derived equation for calculation of concentration of unknown? By ODS into concentration of standard. That is like simple arithmetic equation. ঠিক আছে, মানে এর মধ্যে তো কিছু করার নেই আমার। আচ্ছা, তারপরে next, how to rectify the error of reagent OD? মাইনাস করব, তাই তো? So concentration of unknown will be OD unknown minus OD reagent divided by OD standard minus OD reagent into concentration of standard. And if like volume correction is needed for unit equation, so we have to do the volume correction. ঠিক আছে, মানে যদি আমার স্ট্যান্ডার্ডের কনসেন্ট্রেশন ইজ প্রোভাইডেড ইন মিলিগ্রাম পার লিটার অ্যান্ড উই হ্যাভ টু প্রোভাইড দ্য রিপোর্ট ইন মিলিগ্রাম পার ডিএল, তাহলে উই হ্যাভ টু ডু দ্য ভলিউম কারেকশন। ঠিক আছে, অ্যান্ড হোয়াট আর দ্য পার্টস, এক্সটার্নাল পার্টস অফ colorimeter? External part display the wheel to change the filters, then the place to put the cuvette and a button, start button. And internal parts, what were they? Light source, entrance slit, then condenser. We use a biconvex lens for a condenser, and the use of the condenser is, why we use the condenser? To make the rays parallel. And then comes the filter. Filter is used for making the light monochromatic. And we use what type of filter? Complementary filter. Like the solution color should be complementary to the, or the filter should be complementary to the solution color. Acha, next comes the cuvette. What are the features of a cuvette? It should be 1 centimeter in diameter, ideally should be made up of borosilicate glass, free from scratches and air bubbles. Optically একদম proper transparent হওয়া উচিত। আর flat bottom ও হয়, like test tube গুলো কি হয়, curved হয়, কিন্তু flat bottom হলে better হয়। এটা গেল। আর photo cell, there will be a photo cell and a display।